Olá, pessoal. Desculpem o nosso atraso. Estávamos fazendo pequenos ajustes aqui na nossa apresentação. Gostaria de já anunciar e de lembrar a todos que assinem as listas de presença para eu não ter que interromper o nosso palestrante. Hoje, nós trataremos na parte da tarde, então, sobre a conquista do ambiente terrestre. E esse tema será desenvolvido pelo professor Charles Delwiche, que está aqui conosco. Welcome, Charles. And I will make a presentation, fast presentation, a quick presentation, em português. O professor Charles, ele é professor afiliado do Departamento de Biologia Celular e Moléculas Genéticas da University of Maryland. Ele trabalha, então, com temas relacionados à conquista do ambiente terrestre, a evolução das plantas, também trabalha com a questão da endossimbiose, a origem da endossimbiose nos cloroplastos, a origem das plantas terrestres, as embriófitas, a partir das algas verdes, usando tanto métodos genômicos quanto sistemática molecular, né? E às vezes filogenômica. Tem uma série de trabalhos publicados. Ele é doutor pela University of Wisconsin, né? E ele tem trabalhado e publicado bastante nessas temáticas que nós dissemos. Então, com vocês, hoje nós teremos a apresentação do professor Charles Delwiche. Ok, Charles? Yes, with you.
Thank you, Wellington. It's a pleasure to be here.
Our pleasure, an honor.
As he said, one of my central research interests is understanding how life on Earth made the transition from an aquatic flora to give us a terrestrial flora. And it happens the way that I'm presenting this, I don't have a way to point at things on the screen. But you'll see on the left, there is an image of a desert crust. So microorganisms living in a desert environment. And this is a good analog of what the terrestrial flora looked like before the evolution of embryophytes. You can see there are a number of different colors in that image on the left. Those represent the different pigmentation of different groups of algae that were probably living on land before the evolutionary origin of land plants. But land plants changed everything. And so on the right, we see an image of a flowering plant, and you can see this large and complex vegetation. If one stands and looks out of a window or stands on a hillside and looks across the land, or even if they're in the International Space Station and look down at the terrestrial parts of the world, the vast majority of what you see is a green pigmentation and these large structured organisms that are this lineage that we call land plants. And so they really completely dominate the land, despite the fact that they weren't necessarily the first organisms to come to the land. And the understanding that there is a relationship between algae and plants is not particularly new. This is from a wonderful paper written in 1779, and Ingenhouse made the observation that the matter that grows in water seems to be related to plants. Now, he got it a little bit backwards, so his view was that algae were a kind of plant, whereas the evidence that I'll present to you shows sort of the opposite, that land plants are a kind of green algae. The overall structure today is that first I'm going to talk about what the physical evidence is regarding what the early Earth was like and what its environment was. Then I'll move on to talking about the characteristics of living algae and plants, and particularly we'll focus on the so-called charophyte or streptophyte green algae, those green algae that are most closely related to land plants. And then after we know what the organisms are that are involved, I'll talk about phylogenetics, molecular systematics, and genomics of those organisms, and the information that that gives us about what the relationships are between land plants and algae. And then I'll end with a discussion why this knowledge is important and what some of the practical gains are that can come from this study. We'll take 2 or 3 breaks. I'll probably do a first break either after I've finished talking about the geological evidence or after I've gone through the list of organisms or gone through the diversity of organisms. And please, if you have a question at any time, don't hesitate to ask. So, life has been diversifying on Earth for between 3.5 and 4 billion years. And green algae and red algae represent what is one of the earliest lineages of eukaryotes that can be identified in the fossil record. On the upper right, you see an image of Gripania, which is normally considered to be a eukaryote. That's primarily because it is much larger than typical bacteria and archaea. It's about 1.8 billion years old. And then at a little over 1 billion years, you have Bangio Morpha, where there are images shown in the lower right of this slide. And Bangio Morpha is unmistakably a red alga. In fact, if I found that organism in the ocean today, I don't think I would think anything of it. It's very clearly a red alga. And if you look at the phylogenetic tree shown in the top part of this figure, you can see that red algae and green algae are sister taxa. And that implies that if red algae are well over 1 billion years old, then the green algal lineage must also be of that same age. Although the fossils that we have of green algae are not quite as old as the best fossils of red algae. The whole diagram puts the occurrence of these different organisms into a geologic context. And the lower panel, the tree that's shown in that panel, is just an enlargement of the Phanerozoic, the most recent portion of the tree shown in the upper panel. And the dots that are put onto that phylogenetic tree shows the ages of some of the fossils that pertain to the origin of land plants. And what you can see is that the diversification of land plants began somewhere in the Middle Ordovician. And in the Middle Ordovician, the nature of the fossils are these spores. So there are individual spores or spores that are grouped into clumps of 4. And these are believed to be the earliest evidence of a terrestrial flora. This is well before there are any body fossils of plants. You do find, in addition to spores, there are a few little tubes like the one shown in figure N in the lower right. There are little tubes or fibers, and there also are a few sheets that might be cuticular material. But the majority of the earliest fossils are spores and spore tetrads. And this also corresponds with a significant jump in the Earth's oxygen, with the atmospheric oxygen. There's an early increase in oxygen that followed the evolutionary origin of cyanobacteria, which are the only organisms that are capable of oxygenic photosynthesis. There is also an increase in atmospheric oxygen that probably correlated with the endosymbiotic origin of chloroplasts. But modern levels of atmospheric oxygen were only achieved following the colonization of the land by land plants. And this probably has to do with the fact that you have more burial of carbon when there is lignin or lignin-like materials that decay very slowly. For there to be a significant increase in oxygen, the photosynthesis has to not be countered by respiration of the same materials. So it's when you have burial of photosynthetic carbon that you see a net increase in the oxygen in the atmosphere. And back in 1989, Jan de Vries, who's an expert in this field as well, pointed out this was the year he was born. But in 1989, I published a paper showing that there were complex polyphenolics, materials very closely related to lignin, present in the green alga Coleochaetae. And much more recently, research by William Willatz and his collaborators has shown through antibody-based screening that the materials that are present in the cell walls of Coleochaetae are very closely related to lignin. So lignin was present in algal relatives of land plants, but it was when you began to have large scale, very soon after land plants moved onto land, there was competition for size. And this was probably competition for access to sunlight, but this led to large woody organisms, and when they died, their carbon did not go back into the atmosphere, and so you had a net increase in atmospheric oxygen. Now, the geologic record also indicates some other, sometimes, or maybe somewhat non-intuitive changes that occurred in the physical environment stemming from the origin of land plants. The presence of land plants, they changed the atmospheric oxygen, it changed the silica cycle, which altered weathering, altered patterns of weathering. And I think actually most interesting to me is that it also led to the formation of mud. So mud rock, which is very important in not only determining the nature and texture of soils, but is also important in terms of fossilization. The presence of mud rock in the fossil record increases dramatically once there's a terrestrial flora. So this helps explain why those earliest Ordovician fossils are really just spores, because even the environment to lead to fossilization of body fossils of plants is something which was a creation of a terrestrial flora. And so the evolution of this lineage that I'm calling land plants resulted in a fairly profound remodeling of the terrestrial environment. It led to the conditions that facilitated fossilization, and it set the environmental conditions that permitted the animal colonization of the land in turn. Again, like plants or like algae, there probably were a few animals living on the land prior to the origin of embryophytes, but anything resembling the modern flora or the modern fauna only came around after the occurrence of a modern flora. On the figure on the left is a diorama from the new exhibit at the Smithsonian Institution, and the diagram on the right shows a fossil from the Rhynie chert. And what's remarkable about this is that that's a fossil that's about 400 million years old, but you can see cross sections of plants in there that have superb preservation, anatomical scale preservation of those plant stems. And so in a fairly short time, you went from having minimal terrestrial flora to having plants that have structures that are strongly reminiscent of the structure that you see in modern plants. So once plants had successfully moved onto the land, they diversified very quickly. Now, it's also important to understand, I've already mentioned that there are multiple algae that were living on the land before the origin of land plants. It's also important to understand that multicellularity per se is not unique to the land plants. So there are several multicellular lineages of green algae, and there's also multicellularity within the red algae, both lineages that were present at the time of the origin of embryophytes. And so, although it's tempting to think that it's the structural complexity that made embryophytes, that made land plants a distinctive lineage, there actually were multiple lineages that were multicellular and developmentally complex algae that long predated the origin of land plants. The gray vertical bar shows the Cryogenian, which was one of the periods so-called snowball Earth, when there was global glaciation. And this is 300 million years, 250 or 300 million years before the origin of land plants. So it's not the structural complexity per se that was critical, although that may well have played into it. And as I've already said several times now, green algae have also moved onto land many times. So this is a figure from Louise Lewis and Rick McCourt's 2004 paper showing the multiple colonizations of the land. And interestingly enough, even on the right-hand part of the tree, which shows a representation, or is where we have the streptophyte green algae, even within the lineage that ultimately gave rise to embryophytes, there are several different lineages that have at least some terrestrial members. And so we know that there's something very special about the embryophytes, about this lineage that came to dominate the land, but it's not 100% clear what it is that allowed them to have that dominance. They weren't the only organisms that were present on the land. They weren't the only organisms that were large, complex, multicellular organisms. And yet, they do truly dominate the land. Now, understanding what the close algal relatives were of land plants began to emerge in the 19th century. Pringsheim explicitly talked about the members of the Charophyceae sensu stricto, that is, organisms like Chara and Nitella. These are large, complex, freshwater green algae. They can be several meters long. They have a structural complexity that's strongly reminiscent of land plants. And yet, the way their bodies are organized are very different. They have nodes and internodes that are strongly reminiscent of what we see in embryophytes. But in Nitella and in uncorticated species of Chara, that internode, which might be several centimeters long, maybe as much as 10 or 15 centimeters long, it's a single cell. It does have multiple nuclei in it, but structurally, they're composed of these giant single cells. And so even though the overall morphology is reminiscent of plants, the fine structure is very different. But Pringsheim had pointed out that they were shall we say, suspiciously plant-like organisms. Bauer in 1908 was even more explicit about this, talking about the genus Coleochaetae. And Bauer made note, and I'll talk about this more later in the talk, but Bauer made note of the fact that Coleochaetae has a very plant-like life cycle. And so he proposed that there might be some specific relationship between Coleochaetae and land plants, although I think Bauer thought in terms of Coleochaetae as being more of an analog of the ancestor of land plants than a sister taxon or a what we might call a homologue. Big jump forward in terms of the understanding of the relationship between specific lineages of algae and land plants came in the 1970s and 1980s when people began to do electron microscopy, ultrastructural studies, and to some extent biochemistry of different lineage of algae. And it was observed by Jeremy Pickett Heaps, and this was summarized beautifully in Maddox and Stewart's papers on the diversity of algae. But it was observed that certain green algae had modes of cell division, had flagellar structure and organization, had peroxisomes, and had some other biochemical similarities, including the structure of the cell walls and some attributes of the chloroplast, which were very strongly similar to land plants. And so this led to the realization that there was that there were two great lineages of green algae, one of which I variously call the charophyte or streptophyte lineage. But one of these lineages links together several of these groups of green algae that hadn't previously been recognized as being close relatives of each other, and suggested that they somehow had a close relationship with land plants. The other thing that happened in this same period or a bit later was the movement towards natural systems of classification. Cladistic analysis is one example of that. There are other analytical methods that are more widely used now than cladistics, such as likelihood-based methods. But one of the fundamental observations was that a good classification is a classification that represents the evolutionary relationships among organisms, which can be contrasted with artificial classifications, which are classifications that are based on some arbitrary property of interest, but do not necessarily represent the evolutionary relationships. It can seem strange to say now, but at the time, that is to say, prior to the 1970s, it wasn't altogether clear that the natural classification was the best classification. But the availability of ultrastructural characters and the application of techniques such as parsimony began to lead to an explicit understanding of two great lineages of green algae, with one of those including land plants. So this is now the general structure for our understanding of relationships among green algae. There's a chlorophyte lineage, which includes both freshwater and marine organisms, but accounts for the vast majority of green algal biodiversity. And then there is a second lineage, which consists of relatively few species when compared to the chlorophytes, but that is exclusively freshwater or a few secondarily brackish or salty environment organisms, especially in the Charophyceae sensu stricto. But this is fundamentally a freshwater lineage. Doesn't have very many species in it, but this is the lineage that gave rise to land plants. So I want to spend a little time now talking about what the characteristics are of those organisms. We know that there is this charophyte or streptophyte lineage that gave rise to land plants. I want to focus on them and talk a little bit more about what their specific properties are. I'd love to spend more time talking about the chlorophyceae as well. They have lots of fascinating diversity. There are wonderful things like in the upper right is Caulerpa, which is the image there is the organism is about 0.5 meter long. Those assimilators that look kind of like leaves sticking up into the water are each but that whole thing is a single cell. It's tremendous structural complexity in a single cell. And I also want to shout out on the lower right, Trentopolia, a wonderful terrestrial green alga that's abundant in moist environments, especially in the tropics. But so I wish I had more time to talk about the chlorophytes, but today I really want to focus on the land plant side of this tree. And so I'm going to kind of walk up on the right-hand side of this tree. I'll walk up from the bottom towards the top and introduce some of these organisms. I've also mentioned a few times that there are both the terms streptophyte and charophyte. I use these more or less interchangeably. Maddox and Stewart coined the class Charophyceae, and they talked about it as including all of the algal relatives here, but they didn't in those papers explicitly talk about the close relationship with land plants. And the close relationship to land plants was talked about by Jeffrey, who used the term streptophyte. But Jeffrey, when he talked about streptophytes, used it to refer exclusively to the stoneworts, the Charophyceae sensu stricto, and land plants. And so now both of these terms are sometimes used to refer to the lineage as a whole. I end up using both of them. I wish that I could claim that I was always consistent, but if I'll try and remember to say Charophyte sensu stricto if I'm only talking about the stoneworts. Anyway, mesostigma is one of the most basal branches in the streptophyte lineage. It is a single-celled organism found in anything from little puddles to ponds and small bodies of water in the freshwater environment. And it's a single cell, but one thing that's really striking about it is that its two flagella stick out of one side of the cell. They're not at the top or at the bottom of the cell, but as it's swimming along, they are sticking out of the side. It gives it a very peculiar tumbling motion. And another thing that is fascinating about this organism is that its cells are covered not by cell wall, but by these complex scales. And so on the right-hand side of the figure, you can see at the top a high energy, energy-focused electron microscope, very high resolution electron microscope image of those scales. And then on the lower right, there's a interesting technology. It's called an atomic force microscope that we use to visualize those same scales. And interestingly, the atomic force microscope, that image is actually taken on a living cell. So it's a technique that's able to give you resolution, which is comparable to what you can get with an electron microscope, although a very different view of the surface, but able to do it with minimal preparation of the material. Anyway, mesostigma is a reasonably common, single-celled, structurally pretty simple organism. And then another seemingly much rarer organism that is close to being a single cell. It's little packets of cells contained within the earlier cell walls is Chlorochibus atmophyticus. And it is little clumps of cells, but those cells are not connected together by anything like plasmodesmata. There have been 1 or 2 vague reports of flagellate zoospores from Chlorochibus, but I have never seen the zoospores. I've never seen a micrograph or a clear description of the structure of the zoospores. And sex is unknown. It probably does reproduce sexually, but it's never been documented. And some phylogenies show Chlorochibus and Mesostigma as members of a monophyletic variability, but most of the recent high-quality phylogenetic analyses I've seen show those 2 taxa, Mesostigma and Chlorochibus, as members of a monophyletic group. And then the third basal lineage are the Klebsiormidiales or Klebsiormidiophytes. So Klebsiormidium and Intransia. And these are simple unbranched filaments. There's a genome now available from Klebsiormidium flaccidum. And this lineage is very widespread. There are about 40 named species of Klebsiormidium. I imagine that there are a number of additional species. It's really very widespread, and there are only a few people who've done taxonomic work on it, although there's been some very high-quality taxonomic work on it. It does produce zoospores. Those are not too difficult to observe. Like Mesostigma, it has this characteristic property that the flagella stick out to one side rather than pointing directly up or down. They stick out from one side of the apex of the cell. So zoospores are fairly well known, but like Chlorochibus, sex has not been described. And Intransia is a sister taxon to Klebsiormidium. It has this very distinctive shape to its chloroplasts. They look almost like they're melting. And those 2 paired images in the center of the screen, the thing that I find very interesting about it is that the chloroplast divides before there's any obvious indication of where cell division is going to occur in the cell. So first the chloroplast divides, but you can see if you compare the top image and the bottom one, you can see that where the chloroplast is pinching apart is perfectly aligned with the location of the nucleus. And so it gives you your first visual indication of where the cell plate, or where the furrowing is going to occur. So those 3 lineages, Mesostigma, Chlorochibus, and Klebsiormidium, represent the 3 basal lineages in the streptophytes. And then there are 3 lineages that are higher in the tree. You could call them the advanced charophytes if you want, but these are the ones there are 3 lineages that are more closely related to land plants. So the Charophyceae sensu stricto, or order Charales if you prefer, include Chara, Nitella, also Tulipella, and Lycnothamnus. And I've already talked a moderate amount about these organisms. They're big, they're complex, they're globally distributed. They also have a superb fossil record because in the lower portion of the screen, you can see the zygote or oospore from, in that case, that's a Chara. And the oospore is surrounded by a very thick layer of the resistant polymer sporopollenin. And that means that the oospores persist very well in the fossil record. And in addition to that, although none of these images shows a calcified species of Chara, many of the species do deposit calcium carbonate in the cells. And so that means that they will sometimes form a limestone rock. And so both the calcified body of the alga and the zygote can survive a very long time in the fossil record. There's an excellent fossil record going back well over 300 million years, and a spottier fossil record going right back into the Windy Field chert, which is 400 million years old. So there's a solid 400 million year fossil record for this lineage. It's the only one of the streptophyte lineages that has a really deep it has plasmodesmata and phragmoplastic cell division. So the cell division and the interconnection between the cells is strongly reminiscent of what you see in land plants. It has oogamous reproduction, so both eggs and sperm. The sperm are large, coiled sperm with flagella that stick out to one side, so they're very reminiscent of the sperm that you find in embryophytes that produce sperm. And it's strongly reminiscent of land plants, except for that single-celled organization. There is a genome available from Cara brownii, so this is among the first. I forgot to mention Klebsiormidium also has a genome available. And it's been an important model organism for electrophysiological studies. Another one of the lineages of more derived streptophytes is Coleochaetae. The Coleochaetaceae include both Coleochaetae and Ketospheridium. I have two slides because I'm particularly interested in this order. So this is the genus Coleochaetae. It's composed of branched filaments. It's also structurally very complex, but this is a microscopic organism. The thalli that are shown on this slide range from 100 microns in diameter to 1 millimeter in diameter. I've seen thalli that are 2 or 3 millimeters in diameter, but those are really the largest ones. The filaments themselves are between 10 and 50 microns in diameter. Its filaments grow apically. So it is only the end cell on the filament that will undergo cell division, although filaments that are further back on, or cells that are further back on the filament are capable of differentiation into various structures. They will undergo cell division and differentiation, but they don't give rise to new vegetative growth. So it has apical growth that's also shared with the Caerales. And there is structural complexity for things like the central image shown on this slide. You can see some branching has occurred where the vegetative cells are responding to the fertilization of a zygote, and they will eventually form a layer of cells all around the surface of that zygote. The name Coleochaetae refers to the sheathed bristles. They have a very distinctive structure, and the center image on the bottom allows you to see what one of these sheathed bristles looks like. Like the Caerales, it produces both eggs and sperm. They do have a sporopollen layer around the zygote, but it is much thinner and doesn't have the distinctive structure that we associate with the Caerales. And so while I suspect that these organisms do exist in the fossil record, they have not been described from the fossil record. It has a number of flagellate stages, not only sperm, but also most of the species produce zoospores, and following germination of the zygote, they produce spores that look very much like zoospores, but are meiotic products, and so are called myospores. It's not as diverse as the Charales, although that may simply reflect the fact that not as much taxonomic work has been done. And there are a couple of genomes that are currently in progress for this, for the Charales. I mean, sorry, for the Coleochaetales. And that includes both a genome that's underway for Coleochaete orbicularis, but also a genome that's underway for Ketospheridium ovalis. Ketospheridium, like Coleochaete, is fairly common, has a global distribution, is not very well seen taxonomically, though. It's a widespread organism, but it's small, it's relatively difficult to culture, and it simply hasn't had as much taxonomic or any other kind of research on it. And I particularly point out the Ketospheridium species I found in Puerto Rico, shown in the middle image, the one to the right. You can see the discoidal filamentous structure is reminiscent of Coleochaete. You also can see that it has that very distinctive sheathed hair, similar to the one that I showed in Coleochaete. And you'll often see descriptions of Ketospheridium as if it were single cells or colonies, but it really is a filamentous multicellular thallus. The image to the lower right is one where it visualizes the tube that interconnects these cells. It can be easy to miss that if you don't image it just correctly, but the cells within the thallus are interconnected into a filament. So it is also, like Coleochaete, branched filaments that undergo apical cell division. However, there are no plasmodesmata in the mature cells, while Coleochaete does have plasmodesmata interconnecting the cells. Since both Chara and Coleochaete have plasmodesmata, it seems likely that plasmodesmata have been lost in Ketospheridium. It seems like they should be present at some developmental stage, but Martha Cook has looked at this extensively, and to my understanding, she has never observed plasmodesmata. There is a single description in the literature of sexual reproduction that states that it is oogamous and produces both eggs and sperm, but the eggs are described as having been released from the thallus. I think this is probably correct, but I've never observed it, and the paper that describes it has no micrographs or photographs of any kind. It just has some drawings. I think it's probably correct, but more work needs to be done. It does produce zoospores. I've seen those. And assuming that the description of sexual reproduction is correct, it probably produces flagellate myospores as well. Both Coleochaete and Ketospheridium have oospores that are kind of a little globe shaped. They remind me of the shape of a gourd, and the flagella stick out to one side. So when they swim, there's a very distinctive structure. And then finally, there are the Filamentous Zignamatophyte, or there are Zignamatophytes. Zignamatophytes can be either filamentous or single-celled. Probably the ancestral condition for the Zignamatophytes is a filamentous structure, and probably the single-celled Zignamatophytes are derived. But just last year, there was a report that came out of Michael Melkonian's lab where they described and presented the genomes of a couple of deep branching single-celled Zignamatophytes. And so if that article is the last word on the subject, then the ancestral condition actually is single-celled. This is still an area for research. I'm still holding out for the idea that the ancestral condition is filamentous. Now, Typically, you'll see the Zignamatophytes described as being unbranched filaments, but they actually do have some branching. So the micrograph shown on the left, you can see Fungiosymbiosis with a single basal branch in the filaments. And that's not an artifact, that's not some kind of a mistake. You do actually observe very sparse branching in Fungiosymbiosis, but it's really only that first cell will undergo division, will undergo branching division, and the whole rest of the thallus is unbranched. But the developmental capability for branching is there. The other thing is that there may be a relationship between sexual reproduction in the Filamentous Zignamatophytes and branching, because they form conjugation in the ventral figure. And this is a process which at least involves the differential expansion of one part of the cell wall, which can potentially, or which I think may be developmentally related to branching. But that's really just speculation. I don't have any direct evidence for that other than to say it's a developmental process which is reminiscent of branching. They also have plasmidesmata, although the plasmidesmata are not as, don't resemble plant plasmidesmata as much as those in Chara or Collocae. There have been some polyploid series described from them, so they undergo some complex nuclear changes. There are no flagellate stages known whatsoever. Sex, whether they're filamentous or single-celled, is by pairing of cells, this process called conjugation. And they do not have apical cell division. Chara has apical cell division, Collocae has apical cell division. But patterns of cell division across the filament in the Filamentous Zignamatophytes, cell division is pretty much random where it occurs. So it's not localized in one part of the thallus. There are a large number of species, 4,000 or more species, including desmids. And I'll talk about this a little bit more, but they also have been important model systems. Fungiosymbiosis, in particular, for the study of phytochrome, and Spirogyra has been a model for the study of cell division. And as I've alluded to, there's a huge diversity of single-celled Zignamatophytes. These are the organisms that are called desmids. This is a derived lineage within the Zignamatophytes. Whether or not the ancestral was single-celled, the Desmidiaceae are a derived lineage within the Zignamatophytes. So these single-celled desmids are the derived condition. They're very complex cells. They're single cells, but they have fascinating, complex structural diversity. Microsteria, shown in the lower right, is one of the most famous examples of this. But really, if you look at any of the desmids under the microscope, they're just beautiful, complex organisms. There's no evidence of plasmidesmata. As I said before, there are no flagellate stages, no flagella. They do have very, very complex cell walls, and the genome of Penium has recently become available. So we're beginning to get some genomes out of these organisms. Genome sequencing has lagged, but they are becoming available. And there are also, unlike any of the other charophytes, there are some genetic tools available for the Zignamatophytes. In particular, both Penium and Closterium have been transformed. Abe et al. Used particle bombardment and electroporation to transform Closterium and get nice, stable mutants of Closterium. And Ivan Sorensen and her collaborators used agrobacterium to transform Penium and also reported stable transformants. I have seen more compelling data from Closterium than I have from Penium. So this is still early days of doing genetic transformations, but the techniques are becoming available. I'm sure someone has tried CRISPR, and I expect CRISPR would work, but I have not yet seen any data from a CRISPR transformation of these organisms. Now, so those are all of the lineages of streptophytes other than land plants. And I just want to kind of review, because I said this about each of the different lineages at different times, but I want to point out that the structural complexity that you see suggests that there must be patterns of communication among the cells. So there are almost certainly diffusible growth regulators, basically hormones that help regulate developmental patterning within these organisms. You can see that in the way Coleochaete pulvinata vegetative cells. You also see that in the development and developmental responses in Nitella and other Charales. I've already mentioned the lignin-like materials that are deposited around the zygotes in Coleochaete, but the fluorescence image in the upper right, the red of the fluorescence is chlorophyll, the white fluorescence that you see in the cell walls are these lignin-like materials. Those are deposited in vegetative cells only following the formation of a zygote, and they seem to be responding either to a hormone or to a metabolite produced by the zygotes. And then finally, the conjugation in Zignamatophytes. So the Sparagyra chunkingensis shown in the lower right. But you can see that the cells are able to detect the presence of a compatible strain nearby and then undergo growth in response to the presence of that other cell. So we know that there is signaling which is going on among cells within these organisms. And you also see this complex patterning that results from the differential expansion of cell walls in different parts of individual cells. And so you can see this sequence showing the growth of germlings, in this case in Coleochaete soluta, but there are similar examples from a number of different streptophyte algae where you have preferential growth of individual parts within the cells. Why I think this matters, I'll come back to a little bit later. And I've also alluded to this a few times as I've been speaking, but it's important to bear in mind that these organisms have been used for at least a century as model systems for plant processes. So Mugiotia very famously was used to study the plant light sensing pigment phytochrome. There are these famous studies shown there for the figure from Haupt 1982, where they would shine a micro beam of light through a part of the cell, and you could see the chloroplast rotating in response to that light. And so they examined what pigment was responsible for that response. And the figure that's to the right of that just shows a filament of Mugiotia where you can see what those chloroplasts look in the real organism. There is also, so I apologize for the audio. There's no way for me to turn it off in the web presentation, but you can see the actin and myosin-based cytoplasmic streaming, which is occurring in that Nitella hyalina filament. Really active streaming, and you can see nuclei as they stream past. And so because those are such large cells and have such dramatic cytoplasmic streaming, they were used, especially in the 1980s, as a model system to study the mechanism by which myosin moves along actin filaments. Similarly, the giant cells have made them useful for electrophysiology. They're good analogs for nerve cells. I've already mentioned study of cell wall formation in desmids and cell division in Spirogyra. So they're important model systems, but a lot of this work was done without the realization that they were good model systems because of their close relationship to land plants. And so I'll come back to that theme when I talk about more modern evidence for the phylogenetic relationship between charophytes and land plants. But I think this is a good point to stop, take a little pause, and see if anyone has questions about the basic biology that I have described about these organisms, or about the after this, I'll bring more to molecular biology. So this ends the kind of broad introduction to the organisms. Would anyone like to ask any questions?
Yeah, until now we have not any questions. Until now. But, pessoal, alguma pergunta a respeito? Charles, could we try to reproduce? It was a video about Nutella?
I'm sorry, I didn't quite understand the question.
You show the last slide, the early slide. You show something like a video of Nutella. I don't know.
Yeah,
a movie, but we cannot see the movement here.
You couldn't see it.
Yeah.
Movement.
Could we try to reproduce again?
I can try that again, sure. I'm afraid if it didn't work the first time, it probably won't work again. But now, do you see the full screen?
Yeah, in full screen here.
So look at the image in the lower right. You should see. You can. I hope you can see a cell there. Yes.
Yeah, I can see the cell.
So now you should see movement of the cytoplasm within that cell.
Yeah, yeah. Great. Now I can see that. Just to explain to people. Pessoal, talvez seja melhor ver quando essa cena aí em tela cheia, tá bom? Porque é bem tênue mesmo o movimento. I was explaining for the people to see in full screen also, because it's better. Now I can see that. Thank you so much.
It's very dramatic when you see it. That's in natural time. It's very rapid cytoplasmic streaming. The chloroplasts are by the cell wall and they don't move. So the chloroplasts stay where they are, but the cytoplasm streams past the cell walls. Well, maybe it would be a good idea to take a break of about 20 minutes and people can think a little bit about what I have said and see if anyone has any questions they want to ask before I start talking about the molecular biology.
Okay, 20 minutes?
Yeah, so it's 2:20 now. So it's 2:20 by my time, I guess 1:20 by your time. So we could come back. So shall we say 1:45?
Yeah, okay.
Okay, so we'll resume at 1:45.
Yeah, okay. Actually, here is T3.
Oh, 3:45. Okay, so see you at 3:45. So we're 2 hours. No, we're 1 hour different. Whatever.
Yeah.
3:45.
Okay, thank you.
Okay, see you in 25 minutes.
Okay. Okay, we are back. Delwiche is back. We have some questions before they start, okay?
Good.
The Professor Vedosa asked us, can you read on the screen?
So Spiroglia and Mesotanium are the two single cell that I mentioned, which introduced the possibility that the ancestral condition for the Zignamatophytes is single-celled, not filamentous. They clearly are very closely related to embryophytes, and this is a very important observation. But I think that they are almost certainly still in a monophyletic group with the other Zignamatophytes. So the relationship is the Zignamatophytes as a whole, including Spiroglia and Mesotanium, as that whole entire group is the sister taxon to embryophytes, as opposed to those two particular desmids or those two particular Zignamatophytes as sister taxon. That's a very new discovery, and so additional work will need to be done with them. It's a very exciting observation. So I will. So the question is, have the growth regulators in Coleochaetae been chemically characterized, and are they somehow related to those in land plants? I'll be talking more about that. The specific growth regulators that are responsible for the developmental processes I was talking about have not been characterized, but I will be talking about some plant hormones that are present in some charophytes.
Good. It was just these two questions now, but in the next moment we can pick the others that maybe can surge here, right here.
Yeah, we can continue with this. We'll have, so next I'll talk about the molecular systematic evidence and then try to pull it together to tell us what it means with regard to how this is useful knowledge to understand what the relationship is between these algae and plants. So in 2001, my lab published a paper based on 4 genes. We published a paper where we said that the Charales were the sister taxon to land plants. Now, most of these genes are chloroplast genes, and this turns out not to be correct. But in 2001, we claimed that the Charales were the sister taxon to land plants. And that seemed to make a lot of sense because of a number of very plant-like properties that you observe within the Charales. But at that same time, analyses that were based on larger numbers of genes, and in this case, larger numbers of chloroplast genes, showed a different topology where they put the Zignamatophytes as the sister taxon to land plants and Chara as much more of an outgroup. Now, these analyses were based on very few taxa, and it's always better to have more taxa. But they were very carefully done analyses, and we were very much aware of these at the time. And this provided an indication that the arrangement showing Chara as the sister taxon to embryophytes might not be correct. And we also found in a 2012 analysis, we also found the placement of the Zignamatophytes as a sister taxon to land plants, which was very surprising. we were by no means the first people to report this with nuclear genes. But the really good analysis that I view as a kind of a benchmark analysis for this came from Norm Wickett's lab in 2014. It was a very thorough, very careful analysis of I think it was about 260 genes out of both the nuclear and chloroplast genome, and found really unequivocal for the sister taxon to embryophytes being the Zignamatophytes as opposed to the Kerales. And so I guess I have to admit that I still have some doubts about this, but the best current data place the Zignamatophytes as the sister taxon to land plants. And this is a little bit surprising because if you think back on the structure of those organisms, the Zignamatophytes are single-celled or filamentous. They don't have plasmodesmata associated with them. And they're structurally really don't seem to have all that much to do with land plants, although their biochemistries are very similar, but we would say the same thing of the Kerales and Coleochaetales. So the essential message here is that we have the Zignamatophytes appearing as a sister taxon to land plants, but the branch lengths are very short, and the Coleochaetales and Kerales seem to be of roughly comparable age. That is, they diverged from the lineage very much around the same time as the Zignamatophytes and the embryophytes. And so really, if you're going to think about the properties of the common origin of all land plants, you need to think about the Zignamatophytes, the Coleochaetales, and the Kerales all at the same time. So what then are some insights that we can get from this knowledge? Well, one important one is that Bauer, all the way back in 1908, Bauer had talked about a couple of alternative theories regarding the origin of the life cycle of land plants. And simply understanding that embryophytes are derived from within the streptophytes, well, all of the basal streptophytes have a haplontic life history, with the only diploid phase being the zygote. And so what that tells us is that the origin of the life cycle of land plants must have come from via what Bauer called the intercalation hypothesis. That is, it must be that it was a process by which the zygote underwent cell division and gave rise to a multicellular diploid phase. And so if you look at the image in the lower left corner showing Coleochaete, this is why Bauer talked about Coleochaete as being interesting, because it produces 16 or 32 myospores from each zygote. So rather than having 4 meiotic products, it has a larger number. It seems that the first division is meiosis, and then there are some mitotic divisions, but it's strongly reminiscent of the land plant life cycle. Now that's true regardless of which taxon is the sister taxon to land plants. Another thing that I should mention is that there have been some observations that horizontal gene transfer resulted in the presence of certain genes within plant genomes. So this is from a paper that came out just a few days ago in Trends in Genetics. But Van Etten and Bhattacharya do a kind of a summary of evidence for horizontal gene transfer in eukaryotes. And one of the events where horizontal gene transfer seems to have played a role is in the colonization of the land. So in the lower right-hand corner of this figure, you see the genes that have been proposed as being involved with the transition to terrestrial flora. And there are a couple of genes, one involved with abscisic acid signaling and another one involved with lignin biosynthesis, where it looks like horizontal gene transfer. So there are at least plausible hypotheses that horizontal gene transfer played a role in this whole process, but this is very difficult to interpret because we don't know what the number of genes that have undergone horizontal gene transfer into any of the other relevant lineages is. We don't know what the background rate is of horizontal gene transfer. So more work is going to be needed before we can interpret this, but there do seem to be a couple of genes that have undergone gene transfer. One other phenomenon which is sort of similar to this, and I just realized I forgot to put in a slide to symbolize it, but another important attribute of embryophytes is that most, if not all of them, have some kind of a fungal association, like Mycorrhizae. And so another key factor with regard to the origin of land plants is the mycorrhizal association. But coming back to this matter of the sister taxon, if we assume that the correct phylogeny is one that places the Zignamatophytes as the sister taxon to land plants, the Coleochaetales is the next most closely related lineage, and the Caraphyceae sensu stricto as the sister to that. So this is currently the best phylogeny that we have available. But if you think about the attributes of these organisms, I think a strong way to think about them is as they are all organisms that live in the freshwater environment. And the freshwater environment is by nature subject to much more frequent drying than the marine environment. And so it's unsurprising that members of a freshwater lineage might have independently acquired adaptations to desiccation and drying. And if you think about the attributes of these different lineages, the chlorokybophyte, the chlorokybus, the chlorokybales or chlorokybophytes have been described from soil environments. Klebsiormidium or Klebsiormidiales are soil or freshwater environments, but they tend to be in very shallow environments. The Charales tend to be in relatively deep waters, although I've shown you a couple of micrographs showing them living in quite shallow water, but in most cases, they're in pretty deep, pretty stable water. Coleochaete lives right on the meniscus. And then very interesting, the Zignamatophytes tend to live right on the meniscus of the water or in very small bodies of water. And so I suspect that the distinctive morphologies that we see in the Zignamatophytes and possibly the evolutionary loss of flagellate stages as well may reflect an adaptation to life in very nearly dry environments, sort of minimal sized bodies of water. And so in that context, then it's very interesting to think of the Zignamatophytes and embryophytes as having adapted to the frequent desiccation found in very small bodies of water in different ways, where embryophytes evolved the ability to move water around within the plant thallus. And that's true even for bryophytes that compared to the algae, they have a greater ability to move water around within the plant body. Then it begins to sort of make sense that the Zignamatophytes might be the sister taxon to land plants. Now, regarding signaling, my lab participated in a very interesting collaboration where we looked at ethylene as a hormone using Spirogyra as a model system. And where this work came from is my lab has made extensive use of high throughput sequencing of so one can do genome sequencing, but one can also use the same sequencing technologies used to sequence a genome, but use those technologies to sequence mRNA. And so you get a high throughput transcriptome. And this is a valuable approach because it is easier to work with than a complete genome, but you can get a great deal of information out of it. And so given that we're working with a diversity of organisms, none of which is a well-developed model system, this was a really appealing way to start getting genome scale information. So as of 2014, this data set for streptophyte algae, we had 29 diverse algae, including both streptophytes and chlorophyte algae, but I'm focusing on streptophytes today. And it amounted to about 300 billion nucleotides of data. Each of those libraries assembled into about 20,000 comps. You can think of each one of those as being a candidate gene sequence. And they were pretty long. So the mean length for the libraries was in most cases over 1,000 nucleotides. And even the poor assembly, the mean length was about 667. We got protein coding sequences as long as 5,773 amino acids out of this. So it's capable of providing, this is a molecular motor protein, but we were able to validate this assembly from Klebsiorbidium by comparing it with a homologous plant sequence. So the point is that this transcriptome sequencing can be a really valuable way of obtaining data in a way that is, it's less labor intensive than doing complete genome sequencing. And within this data set, we observed a number of genes that were homologous to genes associated with plant hormones, including sequences that were homologous to the plant ethylene gene. One of the key genes in the ethylene signaling pathway is ETR1. It's the ethylene receptor. This shows a pairwise sequence alignment of the Spirogyra and Arabidopsis ETR1 genes. Well, it happens that within my department, there's a scientist, Karen Chang, who is a leading scholar in the ethylene signaling system found within land plants. And so I took this sequence to her and took the set of plant hormone-like genes that we found and showed them to her. And at first she was pretty skeptical, but when she looked at this alignment of ETR1, she looked at that and could recognize that the active site was conserved, and it really did look very much like a plant ethylene gene. So we engaged in a collaboration to study this. Now, this is probably a good point for me to just remind you of the concept of homology. And so homology is actually a concept that predates Darwin, but in the modern sense, sequences are considered to be homologous if they are derived from the same sequence in a common ancestor. And so it is a statement about the evolutionary origin of the gene it doesn't actually tell you anything about the function of the gene product, other than to say that in a common ancestor, that gene product must have had the same function. But it's possible for a single gene to diverge in function in two different lineages. So yes, we had a sequence that was strongly reminiscent of, or we have a gene that was clearly homologous to the Arabidopsis ETR1 gene, but that doesn't tell us what that gene is actually doing about the evolutionary origin. So we wanted to be able to address the question of functional equivalence. So in collaboration with Dr. Chang, and I'm not going to try to fully explain this, in part because I don't have any convenient way to be able to point at the parts of the figure, but panel A shows you the ethylene signal transduction, so the ethylene perception pathway. And we tested the function of ETR1, CTR1, IN3, and ERF1. So 4 different genes in the ethylene signal transduction pathway. Figures D and E show you that if we take a Arabidopsis mutant, which is lacking the ETR1, the ethylene response gene, and transform that plant with a Spirogyra homolog, so that same ETR1 gene that I just showed you, the Spirogyra gene is almost as efficient at rescuing the deficient mutant as is the native Arabidopsis homolog. So the Spirogyra ETR1 protein functions in an Arabidopsis background the same way as the native Arabidopsis one. And furthermore, if we look at the other genes that I mentioned, these diagrams show IN3, which is a molecule that carries the signal from the endoplasmic reticulum into the nucleus. It also functions in a plant background. And I'm not showing you the data, but CTR1 and ERF1 also were able to show either in tissue culture experiments or by quantitative PCR, also respond in a way that is functionally equivalent to what you see in plants. So it's not just that there are genes that resemble the ethylene pathway, which are present within Zignamatophytes. And I should say these are also, I'll show you in a second, but I should say these are also present in Coleochaetales and Carales. But it's not just that genes are similar to the pathway, but the pathway is actually functionally equivalent. And we were also able to show that Spirogyra does produce ethylene gas, although it does so at very low quantities compared with land plants. But panels L and M are showing the ethylene production. So there is ethylene production, albeit at low rates. And we don't think that the production of ethylene is by the same pathway as ethylene is produced in plants. But so we had shown that the pathway was present and functional and that ethylene was produced. So we had the bioinformatic observation that that ethylene genes seemed to be present. We could show that they were functionally equivalent, but we still didn't know what they were actually doing. We didn't know what the phenotype was. And then an observant postdoc by the name of Bram van de Poel, who's now on the faculty in Ghent in Belgium, but he noticed that in air, the colony of Spirogyra had a different shape than if it was exposed to ethylene. So if you look at panel C, you can see the different structure of the mass of Spirogyra depending upon the presence or absence of ethylene. And what he eventually determined was that ethylene regulates the elongation of the Spirogyra cell. So panel D shows in air versus in ethylene, you see that the cells have elongated substantially. Cell length was the only attribute he could identify which was significantly different between the two. Panel E shows that there's a dosimetric response more elongation of the cells. The superficial look in the image on the screen, it looks like the chloroplast wraps around a different number of times, but that was not significant. And that was not a generalized difference among all of the cells that were looked at. And the diameter of the filament remains unchanged. And then again, as I mentioned, so the response seems to be cell elongation. And the Spirogyra does produce ethylene, but probably not by the same pathway as plants do. And so then the question is, well, if it's regulating cell elongation and it's using ethylene as a response factor, what is its actual biological significance? What is the biological function of this hormone signaling pathway in Spirogyra? And the fact of the matter is we don't know the answer to that. My speculation is that because ethylene is 5 orders of magnitude more soluble in air than it is in water, that ethylene plays a role in signaling to the alga whether it is in contact with the atmosphere or not. And so it's a sensory mechanism that helps it determine whether or not it's growing at the meniscus. That's pure speculation. I don't have any direct evidence of that, but it would make sense in terms of the biology that we see. So ethylene, a plant hormone, previously only known from embryophytes, turns out to be present within the genomes of several streptophyte algae. And at least in the case of Spirogyra, it's not only are the genes present, but they're functionally equivalent. This very complicated figure shows miniaturized pairwise sequence alignments for several other plant hormone pathways. And the arrow points to the Spirogyra ethylene. So that's the one organism where we know that we have functional equivalence. But if you look at these other figures, you'll see that to a greater or lesser extent, there are several other hormones that look like they may have similar mechanisms within the algae. Now, interestingly, there are some experimental data indicating that auxin does function within some of the streptophyte algae, but if you look at the auxin panel, you'll see AUX1, AUX1 seems to be missing from all of the streptophyte algae. And that's actually consistent with what's known about the function of auxin within streptophyte algae, because it does not seem to have the directed polar transport that you see in embryophytes. And then cytokinins look like there may be some similarity there, but much less work has been done on that. Anyway, the important point from this figure is that it's not just ethylene, but it looks like several other plant hormones would be worthy of investigation. A few other studies have been done. So Delwiche et al. In 2012 investigated the function of strigolactones within the lineage. It's not quite as good a match as ethylene. That is, it's not quite as functionally similar as an ethylene, but it does look like strigolactones have a developmental response, at least in the Charales. And then I've already mentioned this a little, but polar auxin transport also. Auxin as a response system seems to be present in some of the streptophytes, but the polarity, the polar transport mechanism is different. So if you look at the Klebsiella in the lower right, you can see that it is fluorescing in response to the auxin transporter, but it does not have the apical in figure A above. The figure in the upper right, panel A, shows the polar distribution. And so now sort of the stage that we're at in terms of understanding the function of these different plant hormone-like molecules, there's a tremendous amount of work that needs to be done to say, because we can now say, well, there are genes that seem to be similar to plant developmental regulators, but we don't really know what they're doing within the algae. Even in the case of ethylene, where we know that it's a functionally equivalent system, we don't really know what it's doing for the biology of Spirogyra. And all of these other pathways should be investigated to determine whether or not they have functional equivalence to the plant system. But we also then, if we know that we have functional equivalence, or in a sense, whether or not they're functionally equivalent, we need to understand what they're doing in algae. And the reason this is so valuable is that it provides a model system, or provides a set of model systems that can help us understand the properties of plant systems, but do it in many cases in organisms that can be grown in a petri dish rather than have to be grown in a garden. And the experiments can be executed relatively quickly. And so it offers potentially powerful model systems which are more nearly homologous to the systems that are present in plants than in other valuable model systems like Chlamydomonas. So in other words, what I'm saying is that by understanding what the phylogenetic relationships are between the algae and plants, it's made it possible for us to better focus our investigations on the organisms that are most relevant to plants. And so consequently, the properties that are observed within these organisms are more likely to be applicable to other problems. So I think that that's a good stopping point. I just want to say that the work that I've been describing here, I've described the work from other labs as well as from my lab, but I want to give a shout out to the people in my lab who worked on this. I particularly want to give credit to Endymion Cooper, who is the person who is in the top center. So at the top of the screen, the image by himself. Endymion did a lot of the transcriptome work and was absolutely a key player in the ethylene project. So that, I think, is a good stopping point, and I would be happy to take any additional questions that you may have thought of. I may have finished more quickly than Wellington was expecting. There you are.
Oops, I'm here. Actually, my internet had some problem sometimes. Here is preparing some storm again.
Oh,
After a warm weather, here we have another storm. Yesterday, I was afraid this storm to close my internet and then have problem here, but it's okay. Oh,
it's amazing technology that we're able to have this conversation a third of the way around the world.
well, another questions, people? I have one question about the characterization. Uh, I'm still, well, the genomic proves until now that the Zignamatophytes is closest to the land plants, and you talk about some points about that. But we have morphological characteristics that resemble a lot of characteristics in land plants also. This characteristic could not be also homoplasy like you cited.
Yeah, so I think that by any interpretation, the Zignamatophytes are highly derived. I'm pretty sure that the reconstruction for the common ancestor would be a branched filament, not a single cell. Okay. So this figure that I showed you, let me see, I think you can see my cursor here. Is that true? Well, anyway, in this figure down in the middle of the tree, there's a little diagram with an arrow pointing to the branch that shows a branched filament. And I'm pretty sure that the reconstruction of the common ancestor of embryophytes, zignamatophytes, coleochaetophytes, and charophyte sensu stricto was a branched filamentous green alga. And so I think whether we're talking about the filamentous or the single-celled zignamatophytes, I'm pretty sure that they have been reduced from a more complex ancestor to that morphology. So this is one of the reasons that I think it's really important to not only think of Spirogyra, but to think of all of these different lineages of charophytes while you're trying to think about what the morphology of the ancestor was. That's my first thought. The second one is that the sister taxon placement of zignamatophytes with embryophytes, this is what we see in all of the multi-gene phylogenetic analyses right now. And so that's the current best hypothesis. That's our current best understanding. But you shouldn't make the mistake of thinking that that's a sure thing. And one of the reasons that I'm very nervous about this is that we expect that when the ancestor of embryophytes moved out of the aquatic environment and into the terrestrial environment, we expect that it was subjected to very different selective conditions. And that probably affected not just things like desiccation, but also things like patterns of mutation, because the exposure to oxygen and ultraviolet light was much higher, and the availability of all the tRNAs probably shifted. And so I'm a little bit nervous that what we're seeing here is parallel shifts in modes of mutation between the zignamatophytes and the land plants. And so I think that some additional analytical techniques will need to be used. That said, right now, the best information we have is that the zignamatophytes are the sister taxon. And that's, I'm rolling with that until we have evidence otherwise.
Good, great. We have another question here.
Yeah, so that's a great question. Are there any mycorrhizae or fungiosymbiosis with algae? I have observed fungal interactions between streptophyte algae and fungi, but never in a way that seemed like a mutualism. I've only seen what looked like pathogenesis. So to the best of my knowledge, there is not any evidence of any kind of a mutualistic association, but it's very clear that the algae do have some developmental responses to their defense responses, but and defend themselves against fungal interaction. And so it seems like the mutualistic interactions are characteristic of land plants, but that would be an exciting topic to try to study more.
Good. Basic question.
Yeah, well, what are the main differences between algae and the terrestrial plants? The overall organization of plants is very different because they have tissue which is composed of multiple cells organized in 3 dimensions. And although we see complexity within streptophyte algae, none of them have the same degree of tissue organization. And then you also have the alternation of generations, so a very different life cycle where there's a multicellular diploid generation, which is why we call them embryophytes, because they're in the streptophytes, they're the lineage that has an embryo, has a multicellular diploid generation. And then you have things that are special adaptations to the land, like the presence of a cuticle, and that sort of thing. But the 2 that I would really point at as being absolutely fundamental for land plants are organization into tissue and the presence of a multicellular diploid generation. Yeah, so the question is, would sporopollenin be an ancestral trait to all streptophytes? And I would say yes, that is almost certainly the case. We have good documentation of sporopollenin in several of the streptophyte algae. Now, there's an interesting thing that in the streptophyte algae, it's typically deposited around the diploid phase, whereas in embryophytes, you find it typically deposited around the haploid phase. But those spore tetrads that I showed you right at the very beginning of the talk, so those early mid-Ordovician spores, they have 2 layers of sporopollenin. They have a big sporopollenin layer and then 4 little sporopollenin layers inside of that. And so it looks like it was deposited around the zygote, and then the zygote underwent meiosis to give rise to 4 haploid ones.
Another substance, could another substance be found also, for example, in algae, for example, hormones and something like a precursor of this kind of substance that have major importance in the riger plant, for example?
Yes, yeah, I think, well, so part of the point that I was making about the hormones is there are complex hormone systems that are present in plants, and some of those systems seem to be present within the algae, but what we don't really know is what hormone systems are present in the algae that aren't present in plants or only partially present in plants. So each one of these lineages probably has its own unique processes, and some subset of those are shared among multiple lineages. And that's some of the work that just hasn't been done yet to really understand the whole picture of how they're working within the algae.
Good. We have a question again from Verusa, Professor Verusa. I would like just to ask her if the question have another part, I don't know, have a second part? Tem uma segunda parte, Vera? A pergunta é essa.
Yeah, so when cells elongate, it can alter the surface area to volume ratio. And so it can have an impact on nutrient uptake and light perception. That is definitely true. And you do find Spirogyra in temperate areas. Actually, I'd be very interested to hear how Spirogyra is doing this in Brazil. But in temperate areas during the wintertime, the cells are very small. And then when it begins to warm up in the spring, they elongate very rapidly. So it's possible that is a response to the high nutrient environments that tend to occur in the spring. But it's also possible that the elongation is associated with actual physical movement, sort of pushing the filaments out towards the atmosphere. And further research is needed to separate these alternate hypotheses for what function that cell elongation is having. We know when in different areas they undergo the cell elongation, but there are several different factors that might actually be driving it. It's a good question.
Another question, people? Well, I think that is good. I was talking here with my ex-advisor. We have an interesting group around here called Serra Dourada. That is an interesting environment in which we can find a lot of filamentous algae, and we have temporary environments, aquatic environments. Then we saw some algae that can develop some characteristics to resist the period when the environment is dry. Maybe we could also expect, for example, that for this kind of groups, we should have similar characteristics for these groups that, for example, that invade the land, for example, characteristics of cell wall and resistance or resistant ways. What do you think about this? We have some homoplasias in different groups that have these characteristics to survive in terrestrial environment, in land environment.
Yeah, this is a very interesting question, and this is interesting, I think, for several reasons. First of all, I wish I knew more about the diversity of streptophyte algae in tropical regions. I've tried to I spent some time studying that. Some of this, or one species of Coleochaete was described from Puerto Rico. And so when I was a graduate student, I spent some time living in Puerto Rico and working with it. But there's a lot more work that needs to be done in tropical regions, both on the diversity of these organisms and what they're doing for their what their phenology is, what their annual life cycle is, and kind of how they're living their lives. This is a very interesting question. But then the thing that you raised about different adaptations to desiccation, like I think of Pithophora or Cladophora, they will often form achenes, so little resistant vegetative cells that have thick walls and resist drying. And many terrestrial algae have a resistant stage where they can deposit a thick cell wall and either tolerate or resist drying. One of the things that I think is very interesting is that there are very few mechanisms like that in the streptophyte algae. Most of them, if they're able to tolerate desiccation at all, the stage that tolerates drying is the zygote. Now, you have desiccation tolerance, that is the ability of vegetative cells to dry out, and then you wet them and they come back alive. You find that in Klebsiella, and you find that in some of the Zignamatophytes. Not, to my knowledge, in the Charophytes, Terraphin, the Carales. And so I wonder if it might not actually be important in terms of why embryophytes evolved from the streptophytes. It may be that they didn't have the ability to form that resistant stage, and consequently there was more selective pressure on the ability to grow in such a way where they could get access to water down in the soil. And so I've wondered sometimes if the absence of those resistant stages wasn't actually one of the answers to that puzzle that I posed right at the beginning of the talk about why is it that this particular lineage came to dominate the land, despite the fact that many other things have transitioned onto the land.
Yeah, it's a good question to answer.
Yeah.
To discover the future of our research.
And the tropical environment is a great place to do some of that research. I would love to have somebody in Brazil describing more species of some of these taxa.
Yeah, just for example here, she said me that the genera that can find in this environment there is Zygnema, Teminogametum, Spirogyra, Edogonium, Zygogonium, just for example, some genera that can be found there.
Yeah, and that's exactly the right taxa. I mean, I bet Coleochaete probably lives there as well, unless it gets too dry. If it gets really dry, Coleochaete won't live there. But a lot of those are familiar algae.
Yeah. For last, I make these questions. I ask these questions for the other research that we had here, and well, I explained this is not a guessing game, but it's just expectations. What do you think that what would be how can I say what will be the future about this kind of studies? For example, in the next 10 years, 20 years, what we should expect for this? And after, how can we, researchers from South America, contribute for this kind of studies? I think that to make some progress in this kind of studies.
Yeah, well, okay, so the first part of that, what do I see happening in the future? Well, I see that we have continuing to have an increasing human population. We have rapid environmental change. We're going into a period where there's really rapid environmental change. Even if we as an international community take very quick and strong action on it, we're still going to see substantial warming of the planet. And the combination of the human population and the environmental change that we're causing means there's going to be a lot of extinction happening. There already is a lot of extinction happening, and it's only going to accelerate. So we're going to see a lot of extinction. So one of the challenges for our generation and the coming generation of scientists is simply going to be to document the diversity that exists and to do so in a very efficient way, because the younger scientists listening to this, by the time they're at their end of their careers, there will have been a tremendous amount of extinction. And every single one of these organisms presents an opportunity for the discovery of useful gene, some of these desiccation tolerance genes, if they're transformed into crop plants, could potentially help tremendously with global agricultural productivity, help us feed people on less land. So I think biodiversity discovery is a very important problem. And I also think, we have to move away from using fossil fuel. So that means that energy systems are going to be more important in coming years. And so understanding the biology of photosynthetic organisms is going to be of increasing importance. So I think both studying biodiversity and studying the fundamental biology of photosynthetic organisms is going to be kind of a growth industry. So that's kind of my response to the first part of that. Now, the question about what can you do in Brazil, in the Southern Hemisphere? Well, Brazil has pretty sophisticated DNA sequencing capabilities. The possibility of doing transcriptomic work in Brazil is very real. You don't have to have a huge team to analyze transcriptomic data. And very little is known about the algal flora in some of these groups. Like I said, I've tried to look at Coleochaeta and Ketospheridium in tropical environments, but I've only been able to do a teeny bit of that. So I think that very significant contributions could be made by a research team in Brazil that wanted to focus on the regional flora. You want to go out into an area that's not very strongly agriculturally impacted. So you need to figure out, where a relatively pristine area is. But then I think by identifying novel biodiversity and getting transcriptomic or genomic data from that, and interpreting it in the context of the kind of questions that I've been talking about, I think there are very real possibilities for Brazil to make important contributions.
Great. Well, thank you so much. We are pleasure to see your presentation and to meet you here. We contact the first time, but I hope that we can make another contact in the future and make another lectures, because I think that this is the first step to provide some innovative themes of research here in Brazil. Then we can expect that in the future we can contact you again.
That would be great. Hopefully, I haven't been to Brazil for a very long time. Maybe sometime, instead of meeting electronically, we could do it in person.
Okay. Thank you so much. Pessoal, muitíssimo obrigado pela participação de vocês. Nós ficamos por aqui. Amanhã nós teremos, então, a apresentação do professor André, aqui da UEL, falando sobre a comunicação científica, né, como que a gente pode transpor isso para a sociedade, e também sobre coleções biológicas. I was explaining that tomorrow we will have here at morning a presentation about scientific communication, not for scientific community, but also for the society. And at afternoon, we will treat about biological collections, and then finish our subject and our course, naturally. Then, thank you so much, Charles, and thank you.
Yeah, you're welcome. Thank you for having me. Bye now.
Bye-bye. Até mais, pessoal.